Okay, plowing toward the end. 10 more slides, not 5. Sorry, I miscounted. Yeah. All right. So the next part of our little story of T cell activation, because this whole chapter is just how do we activate a T cell. So an activated T cell has to go out and do things, and it has to make new proteins, and it has to start dividing. And we've covered a little bit of the stuff that makes it divide, and some of the things that make it do new things. Now let's talk about another big important protein. So mTOR is another one that you might have heard about in like a cell biology context. It's the target of rapamycin. Rapamycin is this, it's like an antibiotic, yeah. And it's also sold as Rapamune, and it's one of the things that they will use to treat people who have organ transplants. Because rapamycin binds to the mammalian target of rapamycin, mTOR, and mTOR is connected. This is one of the most connected proteins you'll see. So it is involved in a lot of driving a lot of cell metabolism, like lipid production, ribosome production, mRNA production, and protein production. Pretty much all the stuff that keeps cells alive. And so you can really strongly affect this in T cells if you treat with rapamycin. You'll basically poison T cells. And by poisoning T cells, they then won't attack and destroy the transplanted organ. That's the whole idea. Yeah, your T cells are, you need them to fight all the other stuff in the world, but you need them to leave your organs alone. So it's a balancing act. You try to knock down the T cells enough that you don't get rejection of the transplant, and you try to leave enough of them on so that you don't get just rampant infection with, various other things. Yeah. So let's go. And so rapamycin ends up being immunosuppressive among the other things that it does in this way, by hitting T cells. So how's that work? Well, where does this mTOR come in? Yeah. Wow. Yeah. Would you believe me if I told you it involved PIP3? So PIP2, think of that as the PIP molecule that can be broken into two parts, an IP3 and a diacylglycerol. PIP3 has an extra phosphate on it. With that extra phosphate on there, it's like a whole total game changer. It's now just a little floating platform that you can attach other things to, and it'll activate those other things. It's like a little key that floats around the interface of the membrane, activating various proteins that bind to it. Yeah. Via that same domain every single time. So let's look. All right. So if you give a mouse a cookie, if you have PIP3 floating along in the membrane, looking great, then PDK is going to bind that. Fine. Sensible. PDK is a kinase. If it's got a K in it, it's probably a kinase. PDK will activate AKT. Fine. AKT is also a kinase, and you can tell from the picture. See that little notch there? It means it's a kinase. AKT will then go and associate with the mitochondrion. Ooh, that's interesting. And it will phosphorylate BAD. BAD is attached to Becal2. Yeah. All right. And so when BAD is phosphorylated, it will then no longer attach to Becal2. Just like with I kappa B, once it gets phosphorylated, it changes everything. It no longer wants to live with its partner. Yeah. Boom. It gets that phosphate and it's out the door. And so once it is released, once BAD is released, it'll bind to a protein that is called 1433. The 1433 protein. Doesn't seem like a protein named that badly could be famous, but this is a quite famous, I believe there are different 1433 proteins. Yeah. Okay. So once that happens, then that is going to promote cell survival. Because you remember these, BAD and Becal2, they were involved in the sort of targets, downstream targets of apoptosis. And so if you take away the BAD from the Becal, then they basically can't destroy the cell. We're defusing a bomb here, people. Yeah. Yeah, we're diffusing a little apoptosis bomb. So this is the survival signal. Remember that T cells get those three kinds of signals, like an activation signal. That's the first stuff we covered. Then a survival signal, yeah, and then they'll also get a proliferation signal via that IL-2. So it'll be more stuff like the things that we covered, but there are multiple mechanisms, because of course there are. Of course there are. So we're going to promote cell survival. Great. But also that activated AKT will now go over here, and in addition to just surviving, it's going to bind to TSC 1 and 2, and it's going to phosphorylate that. And so once it's done that, that will convert Rheb protein, RHEB, into the active form, and it will now no longer associate with TSC 1 and 2, because once they've got their phosphate, they are also straight out the door. Rheb protein binds to mTOR, and now mTOR is activated, and now mTOR is going to go and drive more lipid production, more ribosomes, more RNA, more protein translation, everything, more of everything. That's what you need if you're going to bulk up a cell and maybe get it ready to divide into multiple cells. This is just like standard G1 phase, like let's hit growth as hard as we can, kind of stuff. Driving forward. So that's the thing. That's what mTOR is going to unlock. And are there pathways? You bet there are pathways. Are we going to learn them? No, we're not really going to learn them. So that's okay. We got other things to cover. But yeah, you can look those up in a good cell biology textbook, or you probably had to when you took cell biology. If not, it's out there on the internet. You can go read it. Fine. What about, because when we get activated T cells, remember they have higher affinity LFA1, and they have more LFA1 as well. And so LFA1 is just like ready. It's like, because remember LFA1 wants to grab onto an intracellular adhesion molecule, one of the ICAMs, ICAM1 or ICAM2. Remember, ICAM1 is tall and ICAM2 is small. Yeah. So, ordinarily, our baby naive young T cell has LFA1, and it's in this ordinary, boring, low affinity state. So pedestrian. But when the fishhook comes along, which is LAT, phosphorylated LAT, GADs, and SLP76, that can then bind to ADAP. ADAP can recruit SCAP and REAM, and SCAP and REAM will then activate RAP1, which is going to be one of these GTPases. So they're going to pry it open so it can, get ready and go do something. RAP1 will then take its little GTP over, and it will cause LFA1 to aggregate. So it'll make them form that SMAC. Remember the SMAC? That's the thing in like the center of the little dot where all the proteins are stuck together. They're stuck together because of RAP1, which is activated because REAM activated because it was bound to SCAP, because it was bound to ADAP, so it was bound to SLP76, GADs, and LAT, which were phosphorylated by ZAP70 a million years ago, way back. Yeah, you remember that CD3 stuff? Yeah, that's it. That's the pathway. And it's just a matter of like, learn the pathways. The entire fourth test is going to be the pathways, almost all of it. I'll try and throw, some other question on there for something, just to like give you a little break from it. But yeah, you can see these pathways have a lot of parts, and just focus on the things that are happening on these slides. Write them down. Don't have to draw the pictures. Can draw the pictures if it makes you feel better. Just get them in your memory by any means necessary. Word. All right. Fine. All right. And I think, we're going to do this last little bit, and then that's going to, then we're going to cut off this section, and then we'll go and do one last tiny section. And then finally, Chapter 7 will be behind us. Oh my gosh. It'll be a great day. So, remember old PIP3? It's going to bind to VAV. VAV is going to come along, and it's going to bind to SLP76, which is phosphorylated, which is attached to GADs and LAT, because they always come attached to each other. So this little complex of lat, GADs, and SLP76 is involved in a lot of different interactions. Yeah. Remember back in the day when it activated, or didn't activate, it just held PLC gamma in place so that ITK could activate it. Boom. Yeah. You remember that? Yeah. Of course you remember. Okay. So this is the story of Nick and Wasp. Yeah. Or Knock and Wasp. I don't know. You could call it Knock. I'm going to call it Nick, though. Yeah. Old Nick and Wasp. Best friends. Stuck to each other. But Nick is inactivating Wasp. Oh, no. Wasp could just open up, spread Wasp's wings, and be so much more. If only Nick would get out of the picture. Or whatever. If only Nick would do something. So, what happens here, what Wasp needs is, it needs an active CDC42. And so VAV is going to pry open CDC42 so that we can get rid of the old GDP, put in a nice new GTP. Ooh. And now it'll bind to WASP, and that'll activate WASP as long as it has the GTP in there. And if something comes along and clips off a phosphate, then it'll go back to just falling off, and WASP will close down just like that. And yeah. Anyway, Nick comes in, riding in here on one of these phosphates that was conveniently put there by Zap70. Yeah, how nice for you, Zap70. Nick brings Wasp to the party. Wasp opens up when the CDC42 comes out. Yeah, you bet Nick does, or Wasp does. And once Wasp is open, Wasp is then like a platform that WIP can bind to. And WIP will bind to ARP 2 and 3, and between ARP and WIP, they will start to polymerize actin. And when you polymerize actin, that means you are moving your cytoskeleton. So whereas you and I have muscles that we can move and bones that stay pretty much in place unless something bad is happening to you, and I hope it's not, and I'm very sorry if it did. If a cell wants to move, it actually has to dissolve its skeleton and then rebuild its skeleton over here, and then it will have moved. Yeah. Seems weird, but when you're a squishy bag full of potentially rigid little hard parts that you can polymerize, that's just how things work. Yeah. So this is going to lead to T cells starting to move. They're going to move in a direction. And this can lead to them, like, looking for antigen presenting cells, or finding cells to do things to, just remodeling the T cell so it'll flatten out and make contact with a bunch of different cells. Yeah, a bunch of things that could happen. Okay, do you remember the SMAC? Of course you remember the SMAC, the central part of the SMAC and the peripheral part of the SMAC. And there's a nice picture of it. So, SMAC formation is going to be controlled in part by CDC42, and it's going to be driven in part by reorganization of the cytoskeleton. So not only is it going to change the shape and possibly movement of the cell, but it's going to be necessary to group all those factors together in a CSMAC. And in there, you can have all your T cell receptors, or at least a lot of T cell receptors, a lot of your CD4s, and so since the CD4 is here, like this CD4 is going to have a LUC attached to the tail, and so it could potentially phosphorylate this one and this one. Since they're all together, you can get a little bit of activation turning into a lot of activation. And so you can make room for a lot of ZAP70s to bind, and then when those ZAP70s bind, they can go off and phosphorylate a whole bunch of LAT and a whole bunch of SLP76, and you can get a whole bunch of GADs that goes between and holds those two together to make that fish hook, etc., etc. Meanwhile, you know that that activation pathway led to changes in LFA1 that make it just better at sticking to ICAM1 or ICAM2, for that matter. Yeah, and that's just those are just facts. That's just ancient history. Yeah, it's a lot of terms. I know it's a lot of terms, and ordinarily, if it's my class, I'd spend a lot less time, perhaps, on some of the stuff like the defensins and complement and stuff like that, just so that you can really hammer these pathways, just because they're useful. If you're going to med school, you're going to have to take basically this course again, and believe me, they're going to hammer the pathways there. They're just going to say, yeah, read chapter 7 tonight and be ready, to cover anything about it. Yeah. Nice thing is that most med schools use the Janeway Immunobiology, so hang on to that textbook if you got it. Yeah. It'll come in handy. Okay. That's a good place for us to pause this episode, and we'll do one more last little bit, and that'll finally be the very most last ending part of chapter 7. So tune in.